Welcome to the Explainer. So you know how we've always been told that our DNA is this unchangeable blueprint, like a fixed script that completely dictates your biological destiny? Well, what if it's not? What if the world around you, and get this, even the world your ancestors lived in, is actively editing that script right now? Today, we're unpacking a biological mystery that is literally rewriting everything we thought we knew about human biology. Let's get into it.
Section 1. DNA is not destiny. The identical clone paradox.
Okay, let's dive into this. To really wrap our heads around how our environment shapes our biology, I want you to imagine a totally bizarre scenario. At the exact moment you were born, let's say you had a secret identical clone. But instead of growing up with you, this clone was handed over to a sketchy traveling circus. Now fast forward. Picture yourself 50 years later sitting across a table from them. You guys have lived completely different lives, right? So if DNA is absolute destiny, you should look, feel, and operate biologically in the exact same way. But would you?
I mean, just look at the wild contrast here. On one hand, you've got your life, a pretty normal suburban existence, maybe a highly stressful desk job, and hey, let's say you've been eating Sonic tater tots 5 days a week since 1992. No judgment, but you might be carrying a little extra weight and dealing with some stress. Then, on the other hand, you've got your clone, raised in the circus, professionally riding a unicycle over a tightrope, so they're in crazy good shape. But they were also malnourished as a kid and picked up a heavy smoking habit hanging with the sword swallowers. Now, if a scientist looked at both of your genetic codes, they would see the exact same DNA paragraph, every single letter in the exact same order, and yet, you two look and function completely differently. Why? Because the spaces and the punctuation of that genetic paragraph are totally different.
Section 2. The hardware and the software. Decoding the epigenome.
And this brilliantly illustrates the concept of epigenetics. The word itself literally means above genetics, and it completely changes the message of your DNA. Think of your genome, your actual DNA, as the hardware of a computer. It's the physical machine. But, as we all know, a machine doesn't do anything without instructions, right? That's where the epigenome comes in. It's the software. It tells the hardware what to do. You've got billions of cells in your body with the exact same DNA blueprint, but they rely on that outside software to actually know what their specific job is.
So how does this biological software actually work in the real world? Well, researchers found that one of the main mechanisms is through these things called methyl groups. These are tiny carbon and hydrogen compounds that physically bind to a gene. Their main gig is to provide outside instructions, and they usually do this by telling a gene, hey, do not express. This specific binding pattern is literally how a cell knows if it's supposed to be a skin cell, an eyeball cell, or, a tongue cell.
But wait, there's more. Alongside methyl groups, we also have histones. Think of histones as little protein spools that your DNA winds itself around. It's very mechanical. If the DNA is wrapped super tightly around that histone spool, the gene expresses less. But if it's wrapped loosely, the gene expresses way more.
So, the absolutely crucial point to remember here is this. Methyl groups basically act like on and off switches. They can completely shut down or activate a gene. Histones, on the other hand, act more like volume knobs, dialing the expression of that gene up or down. And every single cell in your body has its own entirely unique pattern of these switches and knobs.
Section 3. The Norbotten generational mystery. Echoes of the 19th century Arctic.
For the longest time, the scientific consensus was that these epigenetic tags were wiped totally clean for a new embryo, basically a fresh start. But then, a massive, fascinating discovery out of Norrbotten, Sweden, completely shattered that idea. It showed us that environmental effects, the actual lived experiences of our ancestors, can be inherited. So Norrbotten is this extremely isolated county in Sweden, located literally inside the Arctic Circle. Back in the 1800s, the farmers there survived entirely on whatever they could grow. Because the climate was so brutal, they cycled between years of horrific, devastating starvation and years of massive, bountiful harvests. And during those rare years of plenty, these starving folks would just gorge themselves. I mean, going absolutely ape-crackers on the food. Fast forward to the late 20th century, and public health specialists decided to look at the birth and death records of the grandchildren of these specific farmers.
And what they found was just staggering. The grandchildren of the farmers who binged heavily during those rare, plentiful years, they died an average of 6 years sooner than the grandchildren of the farmers who went through the starvation periods. 6 whole years.
Let that sink in for a second. It proved, beyond a shadow of a doubt, that bad epigenetic information isn't always wiped clean. Sometimes it gets stuck to the genome and passed down from generation to generation. It means that your grandmother making certain dietary decisions decades ago might have actively programmed the biological software you are running right this second. Conditions we see today, like diabetes, autoimmune disorders, and certain cancers, they might actually be the echoing result of the harsh environments our ancestors survived. It's like a biological time capsule.
Section 4. Hunger, Smoking, and Health. The Changing Epigenetic Landscape.
Now, what's really interesting about this slide is that epigenetics isn't just about what your grandparents did. It's actually a constantly shifting landscape within your own lifespan. Our software is continuously updating based on our immediate environment, our daily behaviors, and, well, just getting older. Look at how our natural baseline changes over time here. Scientists measure DNA methylation at millions of sites across different age groups. A newborn has the highest level. A 26-year-old sits right there in the middle, and a 103-year-old has the lowest. So simply existing and aging naturally changes your epigenetics. But here's the catch. Environmental exposures can radically accelerate or warp this whole process.
Look at these specific triggers. For example, diet is huge. During the Dutch hunger winter famine back in 1944, pregnant mothers were subjected to severe starvation. 60 years later, researchers looked at the babies born from those pregnancies and found they had completely different DNA methylation levels compared to their own siblings who weren't exposed to the famine. This significantly spiked their risk for heart disease, schizophrenia, and type 2 diabetes. And it's not just diet. Smoking severely scrambles your epigenetic tags. High stress literally flips switches in your brain. Even germs use epigenetics against us. The bacteria that causes tuberculosis actually changes your immune cells' epigenetics, flipping an off switch on the IL-12B gene just to weaken your immune system so the germ can survive. It's wild.
Section 5. Reversing the epigenetic damage. Taking back the punctuation.
I know. Hearing about inheriting bad tags or stress permanently changing our genes sounds like a total buzzkill. But here's the incredible, hopeful pivot. We can take back the punctuation. Because unlike genetic mutations, which are permanent, epigenetic changes are reversible. Let's walk through this timeline of a smoker. Heavy smoking causes severe epigenetic damage, right? It shows up specifically as low DNA methylation at the AHRR gene. But look at step 2. The smoker quits completely. Within less than a year, the methylation levels at that gene actually start to climb back up. Eventually, the software rewrites itself, and the former smoker's epigenetic levels return to those of someone who has never smoked a day in their life. Your positive lifestyle choices can physically fix the software.
And as our grasp of the science gets better, it's becoming this incredibly powerful medical tool. We know certain epigenetic changes can spike cancer risks, like how decreased expression of the BRCA1 gene due to methylation raises the risk of breast cancer. But because these distinct methylation patterns show up super early, we can actually use them for screening. Think about commercial tests like Cologuard. They can take a simple stool sample, look for abnormal DNA methylation near certain genes, and help us catch colorectal cancers incredibly early, way before they progress. It's a real game changer.
At the end of the day, epigenetics completely redefines our biological responsibility. Your DNA is not some fixed, inescapable destiny. It is a dynamic, living machine that is constantly taking instructions from the world you expose it to. The choices you make today, what you eat, how you handle stress, whether you smoke, are literally moving the punctuation marks around in your genetic code. And as the Norbotten farmers prove to us, that exact code can echo for decades, maybe even centuries. So I want to leave you with this final thought. Knowing that the biological software you write today could be inherited tomorrow, what legacy are your lifestyle choices writing into your family's genetic software right now?